In this video, we're going to talk about review part 6, DNA, RNA, the central dogma. We'll also talk about mutations. So let's get started. Over here, we have our big category, which is nucleic acid. There's DNA and RNA. ATP is also a nucleic acid, but over here, you only need to remember the two, DNA and RNA. So those are the macromolecules. DNA is very, very long. If you were to stretch it out, it has a lot of information in it. Similarly, RNA also has a lot of information. It's a very long, it's a macromolecule, it's a big molecule. Both of DNA and RNA are made up of nucleotides, but DNA nucleotides are different from RNA nucleotides. But either way, they have a very similar structure of a sugar in the middle, a phosphate group on the side as a little circle, and then we have a nitrogenous base. So for DNA, the sugar will be deoxyribose, and for RNA, the sugar will be ribose, as you can see over here. Okay, and over here is the phosphate group, and we have the nitrogenous base right here. So you can remember it like this. That is one nucleotide. That is the monomer. You will put together a whole lot of these monomers to make an actual DNA or RNA. All right, moving on. This is a labeling type of question. We have number 1 is our phosphate group. Number 2 is the sugar. If it was DNA, once again, that's a deoxyribose, and RNA, it will be a ribose. Number 3 is our nitrogenous base. Number 4 is our sugar phosphate backbone. So in the middle is where the ATCG, AUCG match up as our base pairs, but on the side of the DNA, on one strand, it actually makes up a sugar and phosphate, sugar and phosphate. So what does this look like? We have the sugar, right? The next DNA, when it comes in, really looks something like this. Okay, so that's how they're bonded together. And then we have another nitrogenous base. That we have the sugar phosphate backbone, and then on this side we'll have the base pairing A to T, C to G as an example for our DNA. All right, moving on. We have number 5. Once again, that's our complementary base pair is number 5, and then we have number 6, which is the 5' end, and number 7, which is the 3' end, because DNA is anti-parallel, it runs in 2 different directions. So that's the labeling, as you can see over here is the answer key. You're welcome to pause and take another look at it. Over here, we have a comparison between DNA and RNA. So on the right side, we have our DNA, and on the left side, we have RNA. Generally, there's a lot of RNA that are single-stranded. So you can remember RNA is single-stranded. There are double-stranded cases, but for our purpose, for the final exam, single-strand works just fine. And then DNA is always double-stranded. They can split up during DNA replication, but remember all the organisms that you know of right now, they all have double-stranded DNA. Once again, ribose versus deoxyribose, just the difference in the sugar itself. And then the actual nitrogenous base for DNA, we have ATCG. You can see ATCNG versus RNA is AUCNG. Okay, so that's another difference, U versus T. So you can take a look at this chart over here. You're welcome to pause and take a look yourself. And then when you're ready, unpause and take a look at the answers over here. So this should all make sense. These are things we just talked about so far. The only ones that we didn't talk about are these two. So we have structured into chromosomes for DNA and stores genetic information for DNA, the actual purpose of the DNA. All right, moving on. We're going to talk about transcription and translation. So on the left side, we see a prokaryote. And on the right side, we see a eukaryote. So prokaryote goes through transcription and translation, just like a eukaryote would. But what is transcription? DNA and to mRNA, as in we're making mRNA using the DNA template. That is called transcription. Translation is when you take that mRNA transcript, the mRNA information that you change into a different language compared to the original information of the DNA, and you actually make proteins out of it. Proteins are the ones that do the actual jobs in the cell. The DNA is the instruction, protein are the ones doing all the work, and then mRNA is kind of an intermediate step. So once again, on the left side, we're looking at prokaryotes, and so hopefully you remember at this point, prokaryotes all have cytoplasm and nothing else. It doesn't have a nucleus. Well, it does have a DNA, ribosome, cytoplasm, cell membrane, but it does not have a nucleus. That means everything in a prokaryote happens in the cytoplasm. Okay, so if you're asked a question about transcription, translation, where are you making whatever, it doesn't matter. They all go in the cytoplasm because that's all it has. For eukaryotes, however, the DNA lives inside the nucleus. Right, the DNA is in the nucleus. The nucleus is where, excuse me, all the magic happens. This is where the genetic information, the control center stays. So because you're making mRNA using the DNA information, you have to make mRNA where the DNA is. So that's also the nucleus. And then after mRNA is produced, as you can see on this image, there are two things happening over here. We'll take a little detailed look. There's a 5' cap. There's a 5' cap and a poly A tail being added. And then there's also RNA splicing where you take out the introns. So these are introns. A little hard to see. Change the color. These are introns. Okay, these are all introns. But what we're doing is we're getting rid of them. Okay, and we're adding the 5' cap and the poly A tail. All of these additional steps called RNA processing, those all happen inside the nucleus. And then when it's done, the mRNA will come out of the nucleus and then find some ribosome. These are these big boys, the big molecules, organelles. Those are ribosomes. The ribosomes are in charge of actually making the protein. mRNA has the instruction to make the protein, but they can't just make the protein on their own. They have to actually find ribosomes. And then this one ribosome, as it attaches to the mRNA, it will read it and then just go along and putting one amino acid after the next, and then eventually you'll have a full amino acid chain, and then it will fold into a protein. So this happens inside the cytoplasm, this very last step. But when we're looking at mRNA processing, adding the 5' cap poly A tail to protect the mRNA, or excuse me, splicing out the introns and getting rid of them, this all happens in the nucleus. And then the mRNA, once it's fully processed, it'll go out of the nucleus, go into the cytoplasm. Okay, so you can pause and take a look at this one more time if you need to. All right, moving on over here, it's just the answer key to your review sheet. You can take a look over here, and then let's do another practice. All right, this is not the same one. Okay, let's do another practice. So we have the DNA, ATC, GCC, whatever, right? It's asking you for a complementary strand. So a complementary strand is made when A pairs with T and T pairs with A, C pairs with G, and G pairs with C. Why would you have a complementary strand? You have two strands of DNA, right? And also, if you're going through DNA replication, you have to split out the strand and then add new DNA bases onto here, like onto the original strand, based on the ATCG pairings. It's very important for your cells to know how to do this pairing, and it does. It does a really, really good job. So we have A pairs with T. So I'm starting right here. T pairs with A, C pairs with G, G pairs with C, C pairs with G, C pairs with G, A pairs with T again, and G. Okay, so when you're writing this on the final exam, just be very careful and make sure you're doing one at a time and not messing up one, right? Like it's really easy to turn C into a G, but just be careful with that. And then moving on, let's take a look at transcription. So transcription, it says using the top strand, meaning we're using this particular strand, okay? So we're taking this strand and we're trying to make an mRNA out of this. How do you make an mRNA? When you have an A in the DNA, what it pairs with in the mRNA is a U, because mRNA doesn't have T, right? There is no T in RNA. Normally you do A to T, it's not possible to do A to U. When there is a T in DNA, you can pair it with the A. We do have an A. In the mRNA, C pairs with G and G pairs with C. Okay, so that is our pairing. So let's take a look at how this is done. So I'm going to move over the DNA a little bit so you can see how it's lined up. All right, let's see. So T pairs with A, that's no problem. A normally pairs with T for a DNA, but now we have mRNA, so A pairs with U. G pairs with C, right? C pairs with G, G pairs with C, G pairs with C, no problem over here. T pairs with A, T pairs with A, and G pairs with C again. Oh, see, this is why we read the question. Hold on. It says, my bad. It says to pair with the original. Pairing with the original. Okay, so redo. A pairs with T. Nope, there is no T. Oh goodness. A pairs with U. I'm going from here, okay? So that's the top strand. So the original strand. A pairs with U. There we go. T pairs with A. C pairs with G. So UAG. And then this is CGG, no problem. A pairs with U again. A pairs with U. And C pairs with G. Since this is all made up just for practice, you might notice that when we do translation, you want to use the codon chart, right? And you're using mRNA to codons. So you're reading the chart, right? So you're finding UAG on here, and UAG happens to be a stop codon. Normally it's not supposed to start with a stop codon, but in this case, just for practice, we're taking a look at how you can read the codon chart. So the stop codon is fine. CGG is on the codon chart, of course. We find C on the side, we look over and find G, right? CG goes on here, CGG is right here. It codes for arginine, which is an amino acid. And then we have UUG, we find U on the left side, we look across. This one is UU, UUG is leucine, okay? So these are the amino acid. Make sure you remember, we're going from DNA to RNA and there is no T, it's U. And then when you're doing translation, finding the amino acid, you're always using the mRNA, not the DNA. All right, moving on. Over here, we're taking a look at DNA replication. So DNA replication is you're taking one DNA, right? You have this winding up DNA, but you want to open up the winded up DNA so that you can add the new DNA bases onto the old strand. So in order for us to open up the DNA, to unzip over here, we have to use an enzyme called helicase. So that's your first enzyme. Second enzyme is called DNA polymerase. So we're adding on, we have our original DNA, right? We split up the original DNA, we're adding the new DNA bases. We're doing the A to T and C to G pairing, C to G, for example, G to C over here. Right? We're doing these pairing, but who's doing it? It's DNA polymerase that help us actually make the new DNA. And how DNA polymerase works is that it will work from 5 to 3 and 5 to 3 this way, for example. So that means our old DNA is 3 over here because DNA is anti-parallel, right? So 5 and then 3 and then 5. So what happened over here, I want you to take a look, is if the original is 3 and then 5, right? The new DNA when it's made will have to be 5 on this side and 3 on this side. So you're going 5 to 3 towards the right, okay? Right here. But then on the top, we have 3 and 5, and then we have 5 to 3 towards where it's unzipping. So I'm going to draw an arrow over here. So this helps us understand also the leading strand versus lagging strand. So the leading strand will always go towards the replication fork because it's going in a continuous way. As it's unzipping more, right? We're opening up the DNA. When it's unzipping even more, you can just keep going because you're going towards where it's unzipping. However, if you're doing adding the 5 to 3 away from if you're going, right? We're opening up the DNA. You're adding this way. You open up even more, you have to go back and add more. So the bottom strand is really added in Okazaki fragments, and that is called lagging strand. We'll take another look at that in a little bit. Okay, so that's DNA polymerase. Moving on to leading strand. Here we go. Leading strand and lagging strand. Leading strand is continuous, and you can also see the arrow pointing towards where the DNA is splitting, and the lagging strand is pointing away, and it's in Okazaki fragments. Once DNA replication is its own thing. Over here, we have, actually, we're going to take a look at this diagram first, and then we'll take another look at transcription and translation. Let's take a look at this diagram. This diagram does a good job helping you organize the entire process of transcription and translation. And it's very important for you to take a look right here. So let's go over it. How do we let's find a pretty color. Let's do, I think we'll just use black because there are too many colors on this page, so it's easier to look at. So on the top right here, we have DNA replication, okay? So you can see DNA replication because we're adding on new DNA bases directly onto the original DNA, and we're just opening up like that. And we're adding on like 2 new strands of something, so it can't be transcription either. So we have the black, the blue strands that are winding up, that's our DNA. We have the little triangles are DNA polymerase. So DNA polymerase is the one that's actually adding on to the DNA, right? So it's in this one is 5 to 3 towards and 5 to 3 away on the top. So that means synthesized continuously is on the bottom, and synthesized in Okazaki fragment is the one on the top. So leading strand is on the bottom strand, lagging strand is on the top strand. And then we have the big circle that's doing the unzipping that is called helicase. All right, moving on. We're going to take a look at the types of cells. So right here, we have a prokaryote first. In this prokaryote, we have a cytoplasm inside the cell membrane, right? The shape of the DNA, as you can see over here, is circular, right? It's a circle DNA. And then this process is called, let's take a look at this one first. So this process right here is transcription. And we know that because we're going around the DNA, right? And we're making one stranded thing, and that's the mRNA being made, and that is transcription. And then once the mRNA is done, over here we have something going on, that is our translation. And the items that are being involved right here, we have the green item, that will be our ribosome. And then we have the red item, which is the protein that's being made, okay? And how do you know that? Because we're looking at the mRNA, we're making something else out of it that has to be the ribosome and the protein. All right, some bottom questions. You can find the rRNA. R is ribosomal RNA. So in the ribosome, the red is a protein. It's made up of monomers called amino acids. And lastly, tRNA is involved in the process called translation. Okay, tRNA is the one that is bringing in amino acids to the ribosome to get them all connected. All right, on the right side, we do also have transcription and translation, but we're looking at a eukaryotic cell because we got the outside nucleus, and then we have, outside cell membrane, and then we have the nucleus inside as well. So the shape inside, right, this circle is called the nucleus. The shape of the DNA over here is linear. This process is still, we still have our mRNA right here, right? This is our mRNA. We still have our DNA right here. But then after transcription is done, there's an extra process called RNA processing. So this is, you can write RNA processing. RNA splicing is better. Let's write that because you're cutting out the pieces, right? We're shortening the mRNA, we're getting rid of some part of it. That's RNA splicing. And then the orange part is the 5' cap and poly A tail. Okay, so once this is all done, our mRNA is ready to move out of the nucleus. It does move out of the nucleus, and we have translation going on over here again. So this just organizes all the different concepts of DNA replication, transcription, and translation. Okay. Now let's take a look at 5' cap poly A tail one more time. We got they're added to protect, okay, protect the mRNA transcript before it goes out of the nucleus in a eukaryote. So that's the purpose. Make sure you know that. And then we have mRNA splicing. We're keeping the exons. We're getting rid of the introns. So you also need to do that because the introns don't code for anything. We have the 3 types of mRNA, RNA, and tRNA. Just take a look right here. Pause. Take notes as needed. We got this one. And let's move on to mutations. Okay. Here we go. Mutations. Quick round of mutations. Let's go. So for mutations, we have 2 types of mutations, but overall, whatever the type is, a mutation is when you have a change in the DNA sequence, right? Let's say you have ATTCG, and now it changes to an A, there is no U, ATGG, right? We change one base in the DNA. You can call that a mutation, and you can specifically call it a point mutation because you're only changing one base or one nucleotide of DNA, okay? This will also change one base in the RNA, and then it may or may not, mRNA specifically, and may or may not change the amino acid or the protein Depending on what the change is, you have silent mutation, missense, and nonsense. Let's take a look at that first. This is our original DNA, right? ATG. With a mutation, it changed to ATA, right? It's a point mutation because we did just change one letter, one base, one nucleotide. Normally, with ATG, the original, you're supposed to have UAC. However, now with the mutation changing to ATA, you now have UAU, right? So this is a point mutation. And if you look at the codon chart, you'll see that it's still tyrosine. So this is actually a silent mutation because although there is a mutation, the mutation is there. It has no effect. The amino acid did not change. Your protein will function just fine. Let's take a look at the second one. We have our original DNA and mRNA that comes out of the original DNA. Everything else is not changed, right? The DNA is not 3 bases. It's very long. If there's only one base change, it's still a point mutation. Now, it changes into ATC instead. So the G changes into C. And the C over here changes into a G because we're doing the C, right, to G, DNA to mRNA. Now, in this case, we got a stop codon. A stop codon means you're doing transcription, right? So DNA is this long, but now your mRNA might only be like this long because it stopped right here early. There's a premature stop codon. So we call this a nonsense mutation. Okay, it is still a point mutation, but specifically the type is a nonsense mutation. Our very last one, we have ATG. Instead of changing the last base, we change A into a G, which means this G will go into a C in the mRNA. Okay, and instead of tyrosine, we now have a histidine, right? So we did not change to a stop codon. We did not keep the same amino acid. We did have an amino acid change. This is called a missense mutation. Kind of like you misspelled it, right? There's a little change in the amino acid, just one, not a ton. Versus this one is nonsense. It's really, really bad, right? It's nonsense. Okay, so let's take a look at an example, insertion or deletion. The original DNA is this. If we add an extra A right here, right? I push all of these backward. So now we end up with, we still need to do 3 bases at a time. So A, CG, right? And then GAA, and then A on its own. You can see ACG, GAA, A on its own. The ATT and the ATT in the front does not change. So that's an insertion, right? We are changing, both of these are called frameshift mutation, the insertion and the deletion. And it changes a lot more than just one base, right? So it's not just changing one amino acid. Every single amino acid, like this is going to be a different amino acid. DNA, mRNA is going to change. Amino acid is going to be different. This amino acid is going to be different. And there's going to be a lot more DNA bases behind it. We just don't know what they are right now. All of these, every single 3 groups of DNA is going to go into mRNA. The amino acid is also going to be different. So this changes a lot of amino acid, multiple. And this shifts the entire sequence. Okay, deletion. Let me clean this up a little bit. So if I'm looking at a deletion, we still have our original DNA, right? But in this case, we got rid of this T. So now everything shifts forward. You still need to read 3 bases at a time when we're going from mRNA to protein. So that's why we're grouping it as 3 at a time when we're looking at it. But in your actual DNA, everything is just in one line. Okay, so we have AT, but you have to have 3 letters, ATC now, and then GGA, and then AA again, right? ATC, GGA, AA. And once again, we're changing every single amino acid where the mutation happens and after, okay? And then we have the inversion where things change. For example, over here, we have, instead of CGCA in the DNA, it flips over. So now you have AC, right? Like you're reading it backward. You're reading it backward. ACGT. ACGT, okay? And then we have this one duplicated. Let's say you're supposed to have, just to use it as an example, TGCA. You'll do TGCA and then TGCA again. That's duplication. Translocation involves 2 different chromosomes. These are 2, like this one right here, this is the before duplication, this is after, okay? Versus over here, these are 2 separate chromosomes. You can see they're completely different from each other, and they're doing a little switcheroos. It's like, I have this bottle, I have my head, put my head on the bottle, put the bottle cap on my head. Crazy. And that's called translocation. It's the only one that involves 2 chromosomes. And here's a little comparison chart. Pause and take a look at the answers. Here's the answer. Pause and as needed. And here's another identification of different types of mutation. We already explained it, but this should make sense to you. So pause and see if you remember how to do it. And take a look at the answer key over here as well. Pause as needed. And we're done. That is part 6. Not sure if I'll have time for part 7, 8, and 9, but oh, look, we already did part 7. Oh, goodness. We did mutation. Well, we just have 8 and 9 then. Fantastic. I will see you for the next one. Goodbye.